So today we're going to start with cellular respiration to continue our chapter of microbial metabolism. So in cellular respiration, you have a series of oxidation-reduction reactions that leads to transport of electrons along carriers that ultimately these electrons end in an acceptor of electron. It can be oxygen for aerobic respiration, or it can be another molecule that is sometimes an inorganic molecule and is other than oxygen. So in this case, it will be anaerobic respiration. The point, no matter if the microbe uses aerobic or anaerobic respiration, is to generate ATP. And that ATP is generated through oxidative phosphorylation. Now, some microorganisms are purely aerobic, others are purely anaerobic. So in this case, you call it obligates, and can be obligate aerobe, obligate anaerobe, but sometimes the microorganisms can be flexible in terms of their respiration. So if there's oxygen available, they prefer it, so they undergo aerobiosis, but if it's not available, then they undergo anaerobiosis. And in this case, you call them facultative anaerobes. So in aerobic respiration, we have glycolysis, Krebs cycle, and the electron transport chain. And then, of course, you know that prokaryotes, they don't have membrane-bound organelles, including mitochondria. So they still have the Krebs cycle, just as eukaryotic cells have it in the mitochondria. But in this case, they will have the Krebs cycle going on in the cytoplasm. And then the electron transport chain happens in the plasma membrane, while in a eukaryote happens in the inner membrane of the mitochondria. So in order to transfer molecules that can be utilized by the Krebs cycle to produce this ATP and these carriers of electrons, which is, this transfer of electrons by these carriers of electrons, which are NADH and FADH, NAD and FAD, then you have to convert pyruvic acid into something that can be going into the Krebs cycle. So pyruvic acid is the molecule that is produced at the end of glycolysis, and it will be oxidized and decarboxylated to produce an acetyl group. This acetyl group is going to be bound to CoA, a coenzyme, and then you form acetyl-CoA. And then acetyl-CoA can be combined with the last product of the Krebs cycle, which is oxaloacetate. So as you're converting this pyruvic acid then into acetyl group, you will be losing two carbon dioxide molecules. That's through decarboxylation. And then you will generate NADH. So in Krebs cycle, oxidation of acetyl-CoA produced as it enters into the cycle, it will produce this reduced form of NAD, which is NADH, the reduced form of FAD, which is FADH2, and then you will generate also two molecules of ATP out of GTP, which is basically substrate level phosphorylation. GTP is guanosine triphosphate. So you have one guanosine diphosphate going into one of these steps of Krebs cycle, and then you make GTP, which will have three phosphate groups, and these phosphate groups will be transferred into ADP, so you can form ATP. And as you are going into this Krebs cycle, or tricarboxylic acid cycle, you will be undergoing some series of oxidation reductions, and then as well, you will be decarboxylating some molecules, and then you will be having the formation of CO2 as a waste product. And then again, Krebs cycle is part of the aerobic respiration. So more or less, here is a simplified diagram showing you the Krebs cycle. So pyruvic acid coming from glycolysis will be the carboxylated, forming acetyl group that is going to join coenzyme A to form acetyl-CoA, and then acetyl-CoA will enter into the citric acid cycle. At the same time, you generate NADH. Now, the last product of this Krebs cycle is oxaloacetate. And this oxaloacetate, once it joins this acetyl-CoA, will form citric acid. Citric acid is going to be isomerized into isocitric acid, and then isocitric acid is going to be converted into alpha-ketoglutarate. So you have a dehydrogenase enzyme that is going to remove some hydrogen molecules or atoms from this isocitric acid, and then you will have this carrier NAD plus that is going to grab these hydrogen atoms, these protons, and it's going to be converted into NADH. And at the same time, you will be removing carbon dioxide. So this is going to lead to the formation of this alpha-ketoglutarate. And then this alpha-ketoglutarate is going to be also dehydrogenated. So you will have a dehydrogenase enzyme here, and basically you will be oxidizing this alpha-ketoglutarate to form succinyl-CoA. So this CoA that you had in here lost as you join this acetyl group to form this citric acid cycle now can be used here to form this high energy compound, succinyl-CoA. Succinyl-CoA is going to transfer a high energy molecule, inorganic phosphate, into this GDP to form GTP. And then this GTP is going to, through substrate phosphorylation, transfer this inorganic phosphate into ADP to form ATP. And you will have the formation of succinic acid or succinate. And now, in this next step, you will have the other carrier of electrons, which is going to be flavin adenine dinucleotide, that is going to be stripping off from this succinic acid some hydrogen ions. So it's going to carry these electrons, and it's going to be converted now, this succinic acid, into fumaric acid or fumarate. This fumaric acid or fumarase is going to be, is going to be converted into malic acid, and then you will have the last oxidation reduction step here, in which you will have this NADH carrying hydrogen ions from malic acid and then converting it into oxaloacetate. And then you continue the cycle again. So the intermediate or the sequence of these events with these different molecules, citric acid into isocitric acid, alpha ketoglutarate, and all those can be kind of remembered by a mnemonic. It's like, can I kiss someone for money or as an offer? So that's one way that you can remember it. There's another one that it's a little bit more. Can I keep selling sex for money? Officer? So, but, well, that's part of aerobic respiration, and it's part of obtaining energy. Now, then all of these electrons are carried by NAD and FAD, as FADH and NADH, will ultimately end up in a series of proteins that are found in the electron transport chain. And some of them, they will be proteins, some of them they won't. Like flavoproteins are part of, or FMN is part of these proteins, cytochromes as well. Or ubiquinones, they're not proteins, but each of these participate in transferring electrons. So as you move from the beginning of this electron transport chain to the end, the molecules are more electronegative. So that means that they can attract electrons much easier. So keep the flow of electrons from the beginning of the chain to the end of the chain. And depending on the ultimate acceptor of electrons, whether it's oxygen or other molecule, you will have respiration, aerobic for oxygen, or anaerobic for any other acceptor of electrons, which can be, in some cases, sulfur for microorganisms that uses sulfur, or other molecules. So, as you carry these electrons in this electron transport chain, and this is called oxidative phosphorylation because you will generate ATP, you will also transfer the protons, the hydrogen ions, into the, in the case of the mitochondria, into the inner membrane space. And then you will have something that is called chemiosmosis. So you will accumulate in this intermembrane space between the inner and the outer membranes of the mitochondria, you will accumulate hydrogen ions, and then that is going to create a gradient. It's going to be a concentration gradient because you will have more in the inner membrane space, but also it's going to be a chemical gradient because as you move these protons, the inside of the mitochondria, the matrix, is going to become more negative. And that is going to attract the protons, which has a positive charge, back into this matrix. And how they are going to enter? Well, they will go through the gap in the ATPase synthase. And this is going to form what is called the proton motive force that is going to make the subunits of this ATPase synthase to gyrate, like if it was a rotor. And as it's gyrating, it's going to grab ADP molecules with inorganic phosphate molecules. It's going to join them together to regenerate ATP. And this process of moving these protons from the intermembrane space into the matrix is going to be called a chemiosmosis. And that is going to lead to the formation of ATP. So here we have the movement of electrons through this electron transport chain. So we have this flavin mononucleotide that is going to carry the hydrogen ions and, it's going to carry the electrons and it's going to transfer them into the Q protein. And then this Q is going to transfer them into cytochrome B. Cytochrome B is going to transfer them into cytochrome C1. And then cytochrome C1 is going to transfer them into cytochrome C. Cytochrome C is going to transfer them into cytochrome A. And then cytochrome A is going to transfer the electrons into cytochrome A3. And at the end, you will have oxygen. And then this oxygen is going to bind hydrogen ions that are available in the matrix or in the cytoplasm to generate water. So the flow of electrons then goes in this direction. So as this is happening, you have oxidation reduction. So oxidation is shown in red arrows, reduction is shown in red arrows, and yellow arrows will be oxidation. So then that movement of electrons, as it's passing through these different molecules in the chain, it establishes a proton gradient or proton motive force. Some of these carriers of electrons have specific molecules, like cytochromes. They have iron in them. Other ones, like flamine mononucleotide, well, they derive from flamine adenine. And this is coming from riboflavin, which is one of the vitamins. So microorganisms, they thrive in a host by grabbing iron from them to make their cytochromes. Or the vitamins, they steal the vitamins from the host. Oftentimes, someone who has a serious bacterial infection ends up with anemia because these bacteria fight for the iron to make their own proteins, their cytochromes, or to make their molecules to perform their metabolism so they can live. Of course, the remedy is not to deprive the host from iron so that the bacteria won't thrive. No, that will weaken the host. But we will see that later, we have basically molecules as well that can steal back the iron. So here is what happens in chemiosmosis. So you have this electron transport chain that has some proton pumps embedded within this inner membrane of the mitochondria. So because of its charge, these protons, they have to go through these pumps. And then you will have then the matrix here, and here the inner membrane space. So as these hydrogens move into here, they will accumulate, so that will make a concentration gradient, and then this space, the matrix, will become negative, and then you have two gradients that will move back the hydrogens, the protons, back into the matrix. A chemical gradient in which you have negative space here, attracting positive molecules, and a concentration gradient. So if you have more here and less here, molecules move by diffusion from more to less. In this case, it's going to be passive transport, and it's going to be aided by this pump, the ATPase. So the ATPase will carry back these hydrogen ions into the matrix, and at the same time, these subunits that are like a rotor will bring together adenosine diphosphate and inorganic phosphate, forming ATP. And this is, no matter if it's a prokaryote or a eukaryote, this is where you obtain more ATP. So oxidative phosphorylation in electron transport chain is where you have the most production of ATP. And then here's the illustration about what happens either in the cytoplasm of bacteria and in the mitochondria in eukaryotes. So again, you have these carriers of electrons, which are NADH and FADH. FADH specifically transfer them into Q protein, while NADH into FMN. And as these electrons flow through these proteins, you have as well the pumping of electrons into this space, and then going back, and then regeneration of ATP. And again, aerobic respiration uses oxygen. Deprive anaerobe from oxygen, it won't perform its metabolism, so it will die. So here is the summary of aerobic respiration. You have a glucose molecule and 6 oxygen molecules that will generate plus 38 ADPs and 38 inorganic phosphate that will generate 38 ATPs, 6 molecules of water, and 6 carbon dioxide molecules. In the case of, that's for prokaryotes. In the case of eukaryotes, instead of 38 ATPs, you have 34. The reason for this is because NADH that is generated in the glycolysis in the cytoplasm, it is not transferred into the electron transport chain so that it can carry these hydrogens and then these electrons. It's a little bit less efficient in a way. We have for ATP less than that, but still it's plenty. It's more than what we get in glycolysis. So for carbohydrate catabolism, each NADH can be oxidized in the ETC to produce 3 molecules of ATP, and each FADH can produce 2 molecules of ATP. So for anaerobic respiration, we can have electron acceptors like nitrate, sulfate, and CO3. And it's less efficient to generate ATP than oxygen. Some products of this anaerobic respiration out of nitrate can be nitrites and can be nitrogen gas and water. In the case of sulfate, you can have as products hydrogen sulfide, water. In the case of CO3, you can have this methane gas and water. Commonality, no matter if it's aerobic or anaerobic respiration, is that you generate always as product water. And at least this will be important for the generation, for the cycle of nitrogen and sulfur in our environment. So there are bacteria, for instance, in soil that can fixate nitrogen, and that is useful for plants. And then, of course, for the animals that eat plants, and for us because we eat animals. So it's part of the life cycle. And then you expect to have then a lot of bacteria in the soil that can use nitrogen And then also we have bacteria that lives in environments that is rich in sulfur that contributes to assimilate sulfur available and then help in the sulfur cycle. What is CO3? What is the name of that molecule? Huh? Carbonate. Just leave it that. Okay, so we have here the ATP yield during prokaryotic aerobic respiration of one molecule of glucose. So this is for prokaryotes. Again, it's not for eukaryotes. So in glycolysis, you generate 2 ATPs. In the transition state, you don't generate any ATP. So when you convert pyruvic acid into acetyl-CoA, in Krebs cycle, out of these 6 NADH and 2 FADH, you generate 2 ATPs. So you got 4 up to here. And then in electron transport chain, out of these 2 NADH that come from glycolysis, you have 6 ATPs. Out of the 2 NADHs from the transition step, as they reach the oxidative phosphorylation, you will have 6 ATPs. And then from the Krebs cycle, when you generate these 6 NADHs, you generate 18 ATPs. And then from FADH, you generate 4 ATPs. So if you add everything, you end up with 38 ATPs. So for each of these NADH molecules, you have the production of 3 ATPs. And for FADH, it's 2 ATPs. And then what happens in electron transport chain, you have oxidation reduction reactions. Same in the Krebs cycle, together with some decarboxylations. Okay, so what are the main products of Krebs cycle?
NADH, FADH2, and ATP, as well as carbon dioxide as waste.
Mm-hmm. And then how do carrier molecules function in the electron transport chain? What do they transfer? Electrons. And then what is the yield energy of aerobic and aerobic respiration? Which one yields more ATP? Which one yields less? Aerobic yields more. And then in general, aerobic microorganisms, they tend to grow faster than anaerobic microorganisms. Now let's talk about fermentation. So in fermentation, you have the release of energy from oxidation of organic molecules. This one doesn't require oxygen and does not use oxidative phosphorylation from the electron transport chain. It doesn't use the Krebs cycle. The substrate for fermentation can be pyruvate or a derivative as a final electron acceptor. The derivative can be lactic acid, for instance. And although it's not efficient at all, like it produces very small amounts of ATP, the byproducts of fermentation are important for us. Through this fermentation, we produce For instance, bread, alcoholic beverages. So I smile there. It doesn't enjoy wine, some bread and cheese, for instance, Swiss cheese. So those are products of fermentation. Yogurt, it's a product of fermentation. We produce lactic acid. So here is the overview of fermentation process. So everything starts with glycolysis. And then with this glycolysis, of course, you produce your pyruvic acid or a derivative like lactic acid. And then something that it will be important as you ferment this pyruvic acid or derivatives is that you will oxidate this molecule and you generate out of this NADH, NAD, and that NAD is going to come to glycolysis to carry this hydrogen. So you keep on going with the process. So fermentation is linked to glycolysis. And then if you keep providing glucose, of course, this cycle will continue. So products of fermentation. Well, in lactic acid fermentation, you produce lactic acid, and you can produce lactic acid only. In this case, you call it homolactic fermentation, or you can produce lactic acid and additional products, and you call this heterolactic fermentation. And then pyruvic acid will be reduced by NADH. Lactic acid or lactate in mammals irritates the nerve endings. It produces pain. We have nociceptors, pain receptors, so when you do a lot of exercise, you run out of oxygen, you undergo anaerobic respiration in your muscle cells, you produce lactic acid, and that is what it leads to soreness in the muscles. Alcohol fermentation produces ethanol and CO2. So glucose, of course, will go in the process of glycolysis to be producing, it will produce pyruvic acid, and then pyruvic acid will be converted into acetaldehyde and CO2, and then you will have NADH, which is going to donate its electrons and protons to reduce acetaldehyde to ethanol. And we have microorganisms that are producing this ethanol, like Saccharomyces cerevisiae, it's a yeast. And then you'll have, in this process, as you're producing the ethanol, bubbling, the release of the CO2. So here we have the types of fermentation. So pyruvic acid can either produce lactic acid or ethanol. And again, this it's a reduction process in both cases. So in this case, pyruvic acid will be reduced into lactic acid. In this case, acetaldehyde will be reduced into ethanol. And this is the reduction process, like NADH will be converted into NAD. And I mean, look how poorly efficient is this. It's just generating 2 ATPs. But what we care about is these byproducts: CO2, lactic acid, and alcohol. So here we have a series of microorganisms and what they do with this pyruvic acid. Like we have Streptococcus, Lactobacillus, and Bacillus. They produce lactic acid. So these are homolactic bacteria. The other ones, they undergo heterofermentation. Saccharomyces produces ethanol and CO2, more than one product of fermentation. Propionibacterium, which is a bacteria that we have in our skin, can produce propionic acid, acetic acid, CO2, and hydrogen. Clostridia species, which are basically anaerobic bacteria, it's gram-positive bacteria. This one produces butyric acid, butanol, acetone, isopropyl alcohol, and CO2. And there are subspecies of these bacteria that produces certain products. And then E. coli, Salmonella, they produce ethanol, lactic acid, succinic acid, acetic acid, CO2, and hydrogen gas. Enterobacter species produce ethanol, lactic acid, formic acid, butanidiol, acetoin, CO2, and hydrogen. Some of these bacteria, of course, they are used in pharmaceutical industry or food industry, and some of these products can be used to study the biochemical pathways. Like you can detect acetoin with a biochemical test, and then you can distinguish Enterobacter from any of these gram negatives like E. coli or Salmonella. So this is an eukaryotic microorganism, yeast. The rest are prokaryotes. Some are gram-positive, like Clostridium, Streptococcus, Lactobacillus, Bacillus are gram-positives. Others are gram-negatives, like E. coli, Salmonella. So some industrial uses for the different types of fermentations. So ethanol can be made out of starch or sugar itself, like glucose. Can produce beer, wine. Acetobacter species can produce vinegar. Lactobacillus, Streptococcus, they can produce cheese, yogurt out of milk. Lactobacillus plantarum, out of the cabbage, it can produce sauerkraut. Aspergillus, which is a fungi, out of molasses, can produce flavoring of certain foods like citric acid, it's flavoring. But also citric acid, guys, it's, since it's an acid, can reduce the pH of certain foods, preventing spoilage of foods. Like these potato chips, they use citric acid, not only for flavoring, but also to prevent spoilage of these potato chips. Gluconobacter, it can use sorbitol, this sugar, to produce vitamin C, ascorbic acid. Archaea, like methanol sarcina, as it is fermenting acetic acid, can produce methane, which is a fuel, methane gas. Propionobacterium fiduciaris, out of lactic acid, can produce propionic acid and carbon dioxide. That's what it expands the Swiss cheese, and it gives like this tangy flavor. So, list 4 compounds that can be made from pyruvic acid by an organism that uses fermentation. Ethanol. Mm-hmm. Lactic acid. Mm-hmm. Very good. And this is the overall view of the process of making ATP. Some bacteria or eukaryotes, they undergo respiration, aerobic or anaerobic, while other ones, they use fermentation as a way to produce ATP. And then expect, of course, fermentation to be kind of a fast way to produce ATP, not as much, but it's better than nothing. And at least for us, the byproducts of fermentation is what we use for our daily lives. Now let's look at the use of other molecules for making energy. So not only carbohydrates are used to form energy, but also lipids and proteins. So bacteria have, just like as eukaryotes, enzymes that can break down proteins. So they have proteases and peptidases. As you use these, the proteins will be converted into amino acids that they can be assimilated. Proteins cannot enter the microorganisms directly, so they have to use this enzyme. So this enzyme will be found in the plasma membrane. And once inside the cytoplasm, these amino acids can be deaminated, so you remove the amino group. You can remove the carboxyl group, so this is the carboxylation. And then for those amino acids that they have sulfur groups in it, they will be desulfurized. And then either these modified amino acids can enter the Krebs cycle at different steps to form energy, or some of them, they don't need any modifications and they can enter into the Krebs cycle. And then that's even for eukaryotes. We can have different steps in which these amino acids enter into the Krebs cycle, and then you can obtain energy. So, do you remember which amino acid has sulfur in it? Cysteine. So here is now the view of lipid catabolism. So the enzyme that breaks down lipids is lipase enzyme. And then once lipase acts on the chain of lipids, like the carbons with hydrogens, that long carbon and hydrogens that forms these lipids, you will form glycerol, which is an alcohol, and fatty acids. So, fatty acids will undergo beta oxidation to form acetyl-CoA, and acetyl-CoA can enter into the Krebs cycle, while glycerol will be converted into the hydroxyacetone phosphate, which is part of the glycolysis cycle, and then through this isomerase, this dihydroxyacetone phosphate will be isomerized into glyceraldehyde triphosphate, which once it enters into glycolysis will be ultimately converted into pyruvic acid into acetyl-CoA. It is, well, it wasn't said before, but of course some amino acids also can be converted into molecules that can enter into glycolysis. But then when you move a molecule that is not glucose into glycolysis to generate pyruvic acid, this will be called gluconeogenesis. So here we have an overview of the catabolism of various organic molecules. So proteins will be converted into amino acids that can enter into the glycolysis cycle, gluconeogenesis. Lipids as well, with glycerol, can enter into the glycolysis cycle, and then through glycerol, and then you have fatty acids or amino acids that they can be converted into acetyl-CoA, and then some amino acids go directly into the Krebs cycle. And ultimately, you end up with these hydrogen ions, and then these protons and these electrons going into the electron transport chain. So then all of these are organic molecules. Some microorganisms can use inorganic molecules as a source of energy. So, what is the end product of lipids? Lipids, you convert them into what with the lipases?
Fatty acids.
Fatty acids and
Glycerol.
glycerol. And what is the products of proteins? Amino acids. Yeah, that's simple. Now let's look at how we can identify metabolic pathways in bacteria with biochemical tests in the lab. So, with biochemical tests in the lab, we try to test the presence of enzymes in bacteria. So we have media with a specific substrates that can be utilized by the bacteria that has that specific enzyme, and that will give you a product that is visibly seen. So examples of biochemical tests is testing the carboxylation and deamination, fermentation, oxidation, and production of hydrogen sulfide. So here we have two test tubes that helps to detect the presence of enzymes that makes the bacteria to use proteins. So in this test tube, we have this liquid media, that has a pH indicator. In this case, it's going to be bromocresol purple. So there is certain bacteria that do not have the carboxylase enzyme, so it cannot decarboxylate the amino acids, and it will leave this pH on the media neutral or basic. When the bacteria has these carboxylase enzymes, it will change the pH, so you will have acidity or acid products in the media, and that will change the pH of the media. So from basic or neutral will change to acid, or basically a lowering the pH. So in this case, you will have a change from purple to yellow. So in fermentation test, you will have a bacteria that can catabolize either a carbohydrate or a protein, and then you will produce an acid. And depending on your pH indicator, it is how you will expect to have the change in color. Now, some bacteria not only can ferment this product like proteins or glucose, but also they can produce gas. In that case, you use this inverted tube, like the tiny ones that I showed you in the lab, that is called the Durkheim chamber or Durkheim tube, that if the bacteria produce the gas will be trapped into this tube, and then you will be having the positive test for gas production. Now, some bacteria will have different cytochromes in the electron transport chain, and some of them, they might not have even some cytochromes. So yes, they will have an electron transport chain, but they will be lacking a cytochrome. So for instance, some bacteria don't have cytochrome C, which is an oxidase. And some bacteria have it, like Pseudomonas. Pseudomonas species have cytochrome C, oxidase. So one way to test is by doing this oxidase test. So Pseudomonas will be oxidase positive, and then other species like E. coli will be oxidase negative. So you will have your bacteria either in a test tube, or you put a sample in a glass slide, and then you add this oxidase reagent, cytochrome C oxidase reagent, that it will change the color. So it will give you like a purple color in the case of oxidase positive. Now, other ones may have this enzyme that helps them to use sulfur, and they produce hydrogen sulfide. So example, Salmonella can produce hydrogen sulfide. And then hydrogen sulfide, it's a gas, so that gas can be trapped in a Durkheim chamber, or if you use that, or can be seen in media like iron rich media and sulfur rich media that will have a change of the color from yellow color into dark color. But sometimes you can see just the bubble. Now, Here we have a fermentation test for different bacteria. So this is a media that will have, for instance, a carbohydrate, mannitol, let's say. If the bacteria ferments this carbohydrate, mannitol, the byproduct is going to be a yellow color. It's going to be an acid that is going to change the media into yellow color. Some bacteria will be mannitol fermenters, other ones don't. So in this case, this is a negative test. So in this case, this will be your control. So you haven't grown any bacteria here, and then you have your inverted Durkheim chamber. This bacteria in here doesn't ferment mannitol. And if it doesn't ferment mannitol, it's not going to produce gas. These bacteria ferment mannitol, but doesn't produce gas. While this one ferments mannitol and produces gas. So what is the test that you use to differentiate Pseudomonas from E. coli? Mannitol fermentation, cytochrome B oxidase, the oxidase test. So this one is oxidase positive, this one is oxidase negative. Now for photosynthesis, some bacteria uses light to produce energy. So we're going to see what type of photosynthesis we have to produce energy, which is called photophosphorylation. We have cyclic and non-cyclic. And then we're going to see light-dependent and light-independent reactions of photosynthesis. And we're going to contrast oxidative phosphorylation to photophosphorylation. So photosynthesis, we have light-dependent reactions and light-independent reactions. So in light-independent reaction, which is called dark reactions, you have ATP and NADH that are used to reduce CO2. And this one is reducing CO2 to sugar, so this is carbon fixation. And this cycle is also known as Calvin-Benson cycle. In light-dependent reaction, you have conversion of light energy into chemical energy to form ATP and NADPH. So organisms that are oxygenic, so they use oxygen as part of these photosynthetic reactions. We have plants, algae, and a bacteria type that is called cyanobacteria, or genera. So these oxygenic organisms, they use 6 carbon dioxide molecules, 12 molecules of water, and light energy to generate carbohydrates, water, and oxygen. We have organisms that don't use oxygen as an electron acceptor. These are called anoxygenic. And we have anoxygenic bacteria like purple sulfur and green sulfur bacteria. Typically, these bacteria and cyanobacteria, they live in the oceans. And in this case, for this anoxygenic, the acceptor of electrons is going to be sulfur. So they use 6 molecules of carbon dioxide, 12 molecules of hydrogen sulfide, and light energy to generate carbohydrates, water, and 12 molecules of sulfur. Now, these two, no matter whether they are oxygenic and non-oxygenic, they are photosynthetic. They always use light energy. So we have cyclic and non-cyclic photophosphorylation. So this is the cyclic phosphorylation. So you have, in this case, chlorophyll molecules that upon being hit by light, they can release 2 electrons. And these 2 electrons now move into the electron transport chain, and ultimately you have an electron carrier at the end that is going to help to generate ATP, and then that carrier brings back these electrons to chlorophyll, closing the cycle. In non-cyclic phosphorylation, you have photosystem 1 and photosystem 2, in which you have 2 independent chlorophyll molecules, and you start the process independently as well. So you can have these 2 molecules of chlorophyll receiving rays of light at the same time, and this photosystem 2 is going to release these electrons that are going to go into this electron transport chain. Same for this one. And in this photosystem 1, you will have the carrying of hydrogen ions by this NADP, which is going to be similar as NAD, but it's going to have a phosphate group, and it's going to accept these hydrogens, so it's going to be reduced. It was oxidized, now it's going to be reduced, and then you will have generation of ATP. So this is non-cyclic because just like as this one, these electrons don't go back to chlorophyll. Now in the Calvin-Benson cycle, you have these 3 CO2 molecules as an input, and you will have these CO2 molecules combining with this ribulose diphosphate, which is a molecule that is part of this cycle, and then you will convert this ribulose diphosphate into 3-phosphoglyceric acid. And then you will have 6 ATPs feeding into this cycle, releasing its inorganic phosphate to convert this 3-phosphoglyceric acid into 1,3-diphosphoglyceric acid. And then you'll have NADPH that now is going to be oxidized, so this is a reduced form, is going to be oxidized, adding these hydrogen ions, reducing this molecule, 1,3-disphosphoglyceric acid, so that you will have glyceraldehyde triphosphate. And then this glyceraldehyde triphosphate is going to be the output. And then you will have glyceraldehyde triphosphate that is going to be converted into glucose and other sugars. So, how photosynthesis is important to catabolism?
Produces a sugar molecule that will be catabolized.
And then next question, what is made during light-dependent reactions? In the form of NAD, so what is the end product of photosynthesis?
Sugar, oxygen,
And water. Carbohydrates, water, and oxygen. And how oxidative phosphorylation and photophosphorylation are similar? Phosphorylation is doing what? Adding phosphates, right? So you are generating ATP. In photosynthesis, the organism uses the ATP. And it gives us the oxygen, the carbohydrates, the plants. So basically it cleans our environment and it gives us starch, food that we can use. But they use their ATP. So the ATP is for them and other things are for us. Okay, so going to write a sentence to summarize energy production in cells. So, independently of if it's phosphorylation, photosynthetic phosphorylation, you will have energy sources, like in the case for photosynthetic microorganisms, which will be cyanobacteria, green sulfur, purple sulfur bacteria. You have chlorophyll pigments in the thylakoids that they have. If it's a plant, it will be in the chloroplast. And then you have glucose for some organisms, elemental sulfur, ammonia, some bacteria live of ammonia, or hydrogen gas. So you will have the generation of electrons that will go into these carriers, NADP, NAD or FAD in the case of this side, and then these electrons through these final electron acceptors, whether oxygen or sulfur, you will have the generation of ATP. So if the final electron acceptor is oxygen, you will have oxygenic or aerobic respiration. If it's nitrates, sulfate, that will be anaerobic respiration. And if it's an organic compound, it's fermentation. So those are the requirements for ATP production. So in a sentence, what would you say that how energy it is produced by the cells?
produced by the cell by taking molecules or phosphorylation by light to catabolize the molecule and produce electron carriers, and those go to produce ATP through ATP synthase, and these result in products that are received.
Very good. So everything, it is using the chemical bonds, right? So the 6 carbon molecule of glucose is broken down into 3 carbon molecules. And I mean, I didn't mention this, but in the case of the lipids in beta oxidation, what you do is basically these long polymers of carbons with the hydrogens are broken down into 2 carbon molecules, each at a time. So, I mean, we cannot work with large molecules. We have to break it down. That's catabolism, into small molecules. So now, well, next time, we will be looking at the categories of microorganisms based on what do they use as a form of energy, either carbon or nitrogen, CO2, whatever they use for generate energy. We will see that in the next time.